day, not as cold as it has been. I actually fought my first bout of COVID. I was scheduled to give this talk 2 weeks ago, and Scott quickly stepped in and gave his talk. I participated at home, but a good dose of Paxlovid knocked it out after the first couple of days. So seems like that's my recommendation at least. So we'll go on from there. A few starting points. Let's see. Let me get organized here. We always thank the church and remember the fact that they give us this space to work in. We thank this church community for providing us this space and the emotional space and just the communal space for us to meet and be together. Very important that this happens. I think it supports the goals of the church and they support us as well too. We've made a lot of friends and met a lot of wonderful people here. And uh, that's a very critical thing. Um, I'm just passing through my 12 years worth of notes and pages here, looking for uh, fast thing that I wrote up. Yeah, or get organized before you start talking is really helpful. All right. Um, one thing was uh, a lady asked a great question at the end of the last Science Sunday when I wasn't here, and I don't know if she's here today, but she was talking about concerning sources for good information and support of recycling and sustainability issues and so on. And I think a few people talked to her after that. Is that person here? I didn't see her. She was in the back, but hopefully she's involved in listening at home or whatever as well too. But um, and I think her point was it can be easy to be cynical and give up your ideals when others say, you know, they just threw all this recycling stuff out because they don't even recycle this equipment. I think was her basic point that uh, she wanted to get involved with recycling, and she thought, and her kids were telling her that people just throw their stuff out anyways. And my answer is really, you got to stay positive. You got to find a way to look at the world and say, you know, I can make a difference in it. I can be part of it and I can add to it and add some value. You have to find other people to do that. You have to find other people that are involved and interested. And this is a great place to do that, right? Find other people that support your point of view and that will support you. That's one big reason for Science Sunday. I think one reason I wanted to start this in the beginning, and that Scott and I feel of real devotion to this thing. People who specifically, you know, have know more about sustainability in this area. They're right in this church. Susan Camasto, right, and the whole Green Sanctuary group. Um, Also, Carl Nash, very into. He worked for the EPA for many years. Pat Clancy got involved with the whole prairie system that we have here and so on. So a lot of people in each of their own ways have really gotten involved with sustainability and fixing the environment and so on. Uh, then we had a group named Scarce, maybe exactly what you're looking for. Um, it's been in action for quite a while in the western suburbs. I think the head person is Kay McKean, and she's been here to this church and given some talks at different times. She's a dynamo of knowledge and action and inspiration, and that's really what you want to find, somebody that just you know takes whatever spark of idea you have and helps you fire it up into a flame. So that kind of stuff rejuvenates you. The key is to get involved with others and to learn from them. There's, if you're interested in a website, it's scarceecoed.org, and they've got a whole bunch of people in the western suburbs that can help you out. So I wanted to respond to her, and she seemed to be very concerned about what she can do and have support, and it seems like that's the way. Well, Now I'm looking for my clicker. There it is. I wanted to mention too that it got mentioned at church today, but our dear friend Nancy Fleming, she's a certified nurse and midwife and a PhD, uh, passed away on Friday. And her husband Bob is in the back, came into church, and I know that she's been such an inspiration to Science Sunday, to me personally. She gave talks about the cooking theory of life, how humans learning how to cook food broke down a lot of uh, the starch and the elements in food that made it hard to digest, so that after you eat, you have to sit and rest for about 5 hours while your food digests. And cooking, which is something that we're trying to get back, I think, many of us, right? But how it enabled humans to go on and become more involved in changing the world and starting a whole new process of culture and everything was based on the fact that we can cook food and break down the hard to digest ingredients that way. So she gave a Science Sunday talk about that. I remember her just being another dynamo of involvement and uh, attitudes and ways to change the world and to bring good to the world. One of the stories I remember, she was scheduled. She used to go out on trips for travel across to underserved countries like Nicaragua, I believe, and around in Central America, and give um, uh, work with a group of people that were giving testing for cervical cancer to women. And they'd go out in these far, far reaches up, not to the main cities, but they'd go out into the hinterlands, into the very out of the way places where people were getting no medical care at all, and testing and treating women and young girls too for cervical cancer and saving a lot of lives. And she was such a dynamo that I remember she had one time they were meeting at O'Hare and flying to Nicaragua, I believe this time, and she had kind of a medical question. I think she wasn't feeling well and she couldn't make the plane flight, and she got cleared from the doctor the next day, I believe, or the day after. She got her own flight. She organized her own flight. She flew to the capital city of Nicaragua, got all her arrangements for her car travel, and she went like 10 hours up into the mountainous areas all by herself, to reunite with these people because she knew how important it was to her to be a part of this group. So just somebody that just put so much effort into all she did, and uh, I just uh, loved her to pieces. We were at her house many times, and they'd feed us great meals. We'd have great discussions, and so I just wanted to just announce how much she meant to me and how much she meant to Science Sunday. So um, yeah, thanks for people's presence in my life. I think that means a lot. Another changing. I didn't segue this very well, but yeah. This will segue into my next thing. The apocalypse. Cicada apocalypse is coming in 2024, and the interesting things about it. I don't know. Have you heard about the cicadas coming back? 17-year cicadas. It's called Brood 13 in northern Illinois and southeastern Wisconsin on the 17-year cycle. So that's coming this summer. There are some really interesting videos that you can see if you just type in cicadas and Brood 13 and so on. Um, they live their life. Some are cicadas that come back every year perennially, and some come on all different cycles. So it's a very interesting scientific process to see this all go on. Um, they live most of their life in the ground, right? As pupa, they emerge through the ground. At that point, as they emerge through the ground, a bacteria that's been waiting 17 years, in this case, at least for them, can infect them, and it can eat away their abdomen. And actually, if you start looking, when the cicadas start coming out, you can start looking through and do your own study and see how many actually got infected by this. But it eats away their abdomen and it turns it into kind of mush. You'll know it when you see it, and you can look at it online. But the cicadas come out, and it's a feast for birds and frogs and almost every kind of critter out there. And there's the only way they overwhelm that and actually get to breed again is that there's so many of them, right? But it's a combination of this 17-year cicada apocalypse and the brood 19 or in southern Illinois in 5 southern states is on a 13-year cycle. So you kind of wonder when was the last time this happened, and interesting thing about that is to say that, well, it'd be pretty easy to figure out if one was a 2-year cicada and one was a 3-year cicada, right? You just find the least common multiple of 2 and 3, that every 6 years you'd have them both come out. So obviously the way to do this is to say, what's the least common multiple of 13 and 17? And because they're prime numbers, you basically multiply 13 times 17. Well, 17 times 10 is 170, and 3 more is 21. 221 years ago was the last time both of these broods came out at the same time, which by interest too is 1803, right? 17 times 13 years ago, 221 years ago, 1803, which was the time of what was happening in 1803. Thomas Jefferson was our president, right? We had just bought the Louisiana Purchase, right? Magnified the size of our country by immense amounts. Started the next year, Lewis and Clark expedition. So this was a pretty wild area, all this area, right? I don't think a lot of white people saw the cicada apocalypse of 221 years ago, right? But a lot of animals and birds did, and so on at that time. Right? So if you have a chance, go out and see it and look for that bacteria evidence. It's kind of an interesting thing as well, too. I believe it's, I was looking at. I wanted to check it out again. I'm thinking it's in early or midsummer, but I'm not sure. Maybe somebody can Google that while we're talking and and let us know. To the animals that I just know, I think it passes through their system and gets deposited in the soil, and then it waits another 17 or 13 years for the next group of cicadas to come out, and then goes on from there. So there's no real consequence that I've seen from that. They even look pretty healthy, and the problem is that they can infect other ones. The cicadas that have it, this infestation, can affect other ones. Interesting things about the cicadas too, that the males, you've heard the sounds that can be up to 120 decibels, enormous, biggest sound from any real animal that's really known. But that's the males making the sounds, and what they're listening for. Does anyone know what they're listening for? It's a little click that the female does, so it's like a snap. And if you find a cicada that's walking around that's calling out, it's looking for a female, and you can go to its side, like the side of it, and you click over there, and it'll turn and start walking that way. Okay. Similar thing happens in humans too, right? And then you can walk over here, and it can walk down the leaf the other way for it too. So you can have fun with cicadas. You don't have to just check them for bacteria. That's the more positive thing you can do about them. But yeah, this clicking, very very responsive to that. Okay. Late April? Okay, late April. In back.
Larger population that grows because of they have more food.
Well, I mean, I would surmise. I'm not sure if there's, if there's. I would imagine that there is. I'm not aware of exact studies that have shown it, but animals that eat cicadas are going to be bountiful this time. It's going to be a boon to them. So I'm sure that there'd be, less starvation, less problems, more health and maybe they'll be able to overwinter better. All these things are probably good for them, but it'll obviously even itself out. And later, there's a whole study of the carrying capacity, they call it, of an area of land, and they often do that with deer. You can see that when there's good food and a lot of browse, then the deer really eat it, and the deer population really goes big, and then the fox or the wolf populations or different things that feed on them go as well too. But then it dies off as this browse is all eaten. The next year or that winter, those great amount of deer can starve off and die off, and then the carrying capacity of the land can't support that many animals. So there's definitely a rebound effect from all these things as well too, and I'm sure there is with these cicadas and so on too, and the things that eat on them, feed on them. Okay. All right. So I thought I'd continue a bit better segue. So we're talking about when we talk about the making of the fittest. I need to go back to this. I titled this DNA and the Ultimate Forensic Record of Evolution. And we're going to talk a little bit about facts and evidence and science and so on, but I want to also uh, use that as a basis of talking about DNA and genetic structure and so on as well. I'm feeling like with all that we've gone through, I may not get through everything I want to talk about today, but I think I have the floor in 2 weeks and we'll continue at that time too. But we're basically going to be talking about evidence here. Evidence is a funny thing. Imagine that you have a discussion with a friend and they never knew about this. Can I get through this? They never knew about the America bought a large portion of its current landmass from Napoleon Bonaparte in 1803. Never happened, she says. Well, you do a little research. 828,000 square miles, you say. Portions of 15 states and even 2 future Canadian provinces. Don't believe it, is the reply. America sought to purchase New Orleans and its adjacent coastal lands, just the adjacent coastal lands, just the area right at the bottom in that pink there. That's all America was looking for. New Orleans is obviously a valuable territory to have. Uh, the source for shipping, right, to bring goods from the Midwest now that was growing down the Mississippi River, which you can see is just at the eastern part of the Louisiana Purchase. All this stuff, Abraham Lincoln is known to have taken trips down with goods from this area down to Sangamon River area, down all the way to New Orleans. So America wanted just this small little portion. But your research shows you that the U.S. accepted the offer that France came back with. And France was burdened by war debts, right? At the time, Napoleon and so on. They accepted an offer to pay 50 million francs and a cancellation of debt of another 18 million francs. And total cost of the U.S. was $15 million at the time, which is maybe about $250, $275 million nowadays. You show copies of documents, you show articles from history books, magazines, etc. Even show paintings that were made at the time, right in New Orleans, changing the dropping of the French flag and the flying of the American flag. Right? Then you take a visit to New Orleans with your friend too. You see the preponderance of French cuisine, right? You see the French language that's still in use today. The French names on roads and buildings. You imagine that this amount of knowledge and information would convince almost anyone of the truths in your statement. It would take a pretty stubborn person to continue in their beliefs after all these facts, right? So let's investigate what I want to talk about today. Our body carries information that convincingly supports evolutionary facts. Let's look at a few today with an inquiring, investigative mind. First, I think we need to review some of the amazing discoveries science has accumulated about living bodies. Most of all of what we're going to be talking about today was not known at all by Darwin in his day or other people 150, 200 years ago. Not known. Okay. Our bodies, as well as those of all living things, are made up of cells. There are 37.2 trillion cells in humans on average. Almost all types of cells have a nucleus, and in any nucleus of each cell, there's contained all the information to build another you. Darwin didn't even know that, right? We're past where Darwin was, and he still came up with some amazing ideas. I think that's fantastic to know. The ribosomes we've learned a lot more about since I was in grade school and high school. What their function is in the cell. So amazing stuff. So the location of the DNA in a cell, human cells, the nucleus is shown down there. This is greater and greater magnification you can imagine. In the nucleus are contained the chromosomes. In the human, there's 23 pairs of chromosomes, right? In chimps and other primates, there's 24. We've known that now that's because we've blended together two of the chromosomes in chimps to give us only 23 pair. So all this stuff we know. Okay. And inside the chromosomes, on these chromosome pairs reside the genes that carry the coded information that your cells use to manufacture all the structures in your body. So this is a tighter in and tighter in view. Again, I think this is a nice thing to just think about. Maybe some of you, this is all a bit of a review and a look at, but in another sense, it's an amazing thing to think about how we've come to this point in really the last 50, 75 years. If unwrapped, these chromosomes spirally wound and tightened and balled up in ways that we're finding out now are very complex and intricate and important too. From only one cell in your body, the chromosomes from only one cell in your body would stretch out 6.5 feet long. In the 37.2 trillion cells in your body, each one has chromosome knowledge that would stretch 6.5 feet in length. That's taller than anyone here, right? Stretch far past the moon. That's the potential for a lot of information. The information is kept on the famous double helix, modeled by Rosie Franklin, James Watson, and Francis Crick in 1953. Actually, deoxyribonucleic acid was discovered in the 1860s. I think that a man at that time was looking at white blood cells in pus, and he found deoxyribonucleic acid, but they didn't know what it did. It wasn't until the 1930s, I think, that they saw that it had anything to do with heredity and reproduction. So this is that famous double helix modeled by Rosie Franklin and James Watson and Francis Crick. It's deoxyribonucleic acid, and it consists essentially of long strands of coded instructions that specify the order of each cell's manufacture of proteins. Proteins are the molecules that do all the work in every organism. Proteins tell your body what to do, tell the cells what to make. From carrying oxygen to building tissue to copying DNA for the next generation. DNA is made of two strands of four distinct bases. Now, some of you probably know this, represented by A, C, G, and T, adenine, thymine, guanine, and cytosine. When DNA splits apart and starts making RNA, then the T, thymine, is replaced with uracil on RNA. Okay. But there's two strands, and it's always A always pairs with T, and C always pairs with G. And the importance of that is it causes uh, perfect, with the exception of mutations, replication, right? When these things split apart, and then an RNA uh, molecule is formed alongside it, then it has perfect transcription of the proper molecule. So it passes on information from generation to generation to generation. So each of these, this would make an enormous book with information on it, right? The, the DNA text would look something like this: all A's, C's, G's, and T's for hundreds and hundreds and hundreds and hundreds of pages, right? It's phenomenal, okay? Question? Maybe we can get it to.
I read that. I've looked at those and I don't understand how they're, how do you read that? Those strings, A, G, T, T.
Okay, that's the next step. We're going to talk about that. All right. It's a code, right? And you have to break a code. So this is telling the body to make proteins. This is telling that cell to make proteins, to line them up in a pattern to make proteins. And there's a pattern to this in some way. And that's been the work of the last 35 years, I'd say, breaking the pattern and understanding it and so on. And I'll give a simplified answer to it. I don't think I know it all, and I don't think humanity knows it all yet, but we've worked towards a lot, and it's amazing what we know. And how all of this, again, supports Darwin's theory of evolution, and that's, if we have time, we'll get to that today too. So this would be a single strand after the DNA splits apart during mitosis when cells replicate and make new cells. Then an RNA strand comes along and it's coded, transcribed to build up the base, the nucleotides they're called, that are the G, C, U, and A on the RNA, and it makes these codons. 3 bases in a pattern make a codon, and each codon stands for one of the amino acids that help make the proteins in your body. So if you look at the first one, GCU there, that's a code for alanine. The next 3 are ACG, and that's threonine, and so on through that whole path. And it's critical, any mutation in here would change it. If one of them drops out, it just gets read the next 3. It would complete, just one drop off in this code transcription would change the whole code pattern, right? It doesn't have the capacity that you and I can look at the word TH blank N and say, oh, it's then or thin or something, right? But it doesn't have the capacity to do that. It reads the next 3 and it would change it to a different protein, and that would be a mutation that would be a change in the production of proteins for that cell. It may be good. If you look at the odds, it's probably not so good, right? But those changes are critical. That's what makes evolution occur, actually. Okay, so you can see that last one, UAG, is actually a stop. It stops that coding process. And at that point, there may be up to 400 strings of amino acids that call to make a certain protein. Does that kind of answer your question? Okay, all right. It's a simple answer. So proteins are made up of building blocks called amino acids. Each amino acid is encoded as a combination of 3 bases, or a triplet: ACA, GDT, GTT, etc. 400 on average amino acids assembled in a chain specify a protein. Okay, so when there's 400 of those, now you got a protein. And the length of DNA that codes for a protein, you probably know that, it's called a gene. Okay, so that's what a gene is in your body. There can be numerous genes on chromosomes, right? Stopped and started and so on. And it's also included with, beyond just this coding stuff on there, there's also what has been called in the past and is probably not looked on as fondly anymore. You've heard the word junk DNA, stuff that's non-coding, right? That doesn't really code properly. And why is it there? That's been an effort of a lot of discussion over the years. There's a whole variety of things that people are experimenting or trying to understand about it. Okay, so now you've got these different amino acids being made in a process that combine to make a protein that tell the cell something to do. But actually, if you think of it, there's 64 combinations, triplets, AAA, ATT, ATC, ATG, all, okay? There's 64 possible combinations. You get that because there's 4 in the first set, in the first triplet, 4 in the second nucleotide set that make a triplet, and 4 in the third. 4 times 4 is 16. 16 times 4 is 64. So there's 64 mathematically possible combinations. But there's only 20 amino acids. Okay? And that's critical and important and helpful. Okay? Multiple triplets code for certain amino acids. And 3 triplets code for nothing and act as a stopping point for protein manufacture. So if you think of the 64 and you take away the 3 stop, you have 21 making 60 or 60 possible triplets, 61 triplets making 20 amino acids. There's roughly 3 for each, right? Some only have 1, some we'll look at a little bit later have 6. Okay. So the stopping points cause they stop the protein manufacture. Now, from all we've said so far, would you say that these codes, ACA, all of a sudden are the same, or do you think they vary in different between species? Are these codes in all species, or do you think they're different in different species? Okay. It wasn't long ago that it was thought by the top biologists that it had to be different for each species. That's what makes things different. But it's the same. In fact, do they vary more, do you think, in things like maybe a tomato compared to a human as compared to a chimp and a bonobo or something like that? No, these things stay the same. They're the same codes through all of life, which is an amazing thing. Not only tomatoes and chimps and bonobos and so on, this is the same code that's used in plants, in bacteria, in archaea, things that diverged from us 4 billion years ago, 3.5 billion years ago. It's a phenomenal, phenomenal thing that life is all created along this same pattern. Okay? With very minor exceptions, exactly the same in every living animal, plant, bacteria. And that's why those of you that maybe or know people or have issues, for example, diabetes, human insulin production used to be when they first started finding out that insulin was what stopped people from diabetic reactions, they had to get insulin from dogs and inject it into people and so on. And it was a very painful process for the dogs and not very pleasant and not easy to cultivate and hard to get enough. But now this shows the process in which we use nowadays. Eli Lilly in 1978 developed the process to make human insulin in bacteria, okay? So there's a bacterium here in the bottom left. Actually, they make it with E. coli, which is pretty prevalent. And they can cut out a certain part of the DNA in that next step there. Does this thing have a pointer? All right, it's got me baffled. from the top, from human pancreas cells, they can take the human insulating producing gene, the portion of our chromosome that actually produces human insulin, and they can add it to an E. coli DNA package, okay? And they can introduce that into a DNA, into a bacterial cell. You can see bottom is kind of representing a large vat that they'd be culturing this stuff in, it would be growing in, and that stuff would grow and reproduce and make more and make more. And then you have in this picture right here, vast amounts of bacteria containing human insulin in it, which then they can extract and purify it and then produce and give you insulin now for $35 for anybody for a month's supply, I believe. Okay. So because the structure, the coding, everything we've talked about how genetic pairing and reproduction occurs, you can use E. coli to make human insulin and be a very time cost savings kind of method. Okay. So just repeating, another item of importance to note is that a large portion of DNA, like I said earlier, is non-coding. It doesn't direct the cell to make a protein. In the past, we called it junk DNA, but scientists continue to see how these strings have other important functions too. Some are in just the way that the string is combined, the chromosomes are twisted around and so on. Sometimes just the space in there causes one section of chromosome that may be linearly, if you did stretch it out in a linear pattern, it would be very far from something else. They're seeing that some of these things automatically coil back in an important place. They cross over in a place where they can have some impact on another part of that same chromosome. So that junk DNA takes up important space to give it geometry, a configuration to this chromosome that allows it to properly match up and do its work. Okay. But other functions are other functions of it are switches that turn things on and off, that start things going. This is kind of found in what they call the evo-devo part of science, evolution and development things. How as a chick embryo or us, and a human embryo grows, at certain times, certain proteins are turned on to cause finger development or lung development or different developments of different parts of the body. And then they're turned off. These switches turn on and off at certain times. So even though the DNA doesn't code for a protein, it acts as a switch and it allows certain things to occur at proper times. So the whole, it's like a symphony of development and construction of the body. So amazing, amazing stuff. And we're just starting to learn some of that. It used to be called, 30 years ago, it was familiarly called junk DNA. So, but all this is kind of a hopefully some level of review of cell structure and so on, and some level of interesting new stuff that maybe you hadn't heard of before. But we haven't really been talking about evolution yet. Okay? But let's go there a little bit more. Evolution is caused by mutations. This is something I alluded to a little bit earlier. The code carried in DNA is always subject to mutations. It can occur by chance. It can occur by some level of nuclear kind of infiltration of things. A whole bunch of varieties of things can cause mutations. Here's one of the amino acids. We'll kind of delve down deep and use this as an example. This is leucine, an important amino acid. And we can start from the original triplet. The 3 codes, TTA, code for the cell to produce the amino acid leucine. But sometimes something can occur that by chance or by other occurrences, something can happen and change This is a mutated triplet. If the A is mutated and is replaced with a G, it'll still code for leucine. Okay. Again, if you take that original TTA and change the first element of it, CTA, it still codes for leucine. Here's a double mutated change. The T could change to a C in the beginning, and the A could change to a T, and you still have leucine. Okay. So those are some interesting possibilities. Again, another triplet. If there's a C and a T, even a C would work too, not just a T. And actually, a C and a T and a G would work as well. 6 different ways for your body to produce leucine. Okay. You wonder why is that, right? It's important. Okay. So what do all these mutations do? Any other mutation would cause another amino acid, right? Or not only that, but if there just didn't happen to be one of these call outs, if one of those codons, one of those nucleotides, one of those 3 letters was not there, that would cause a whole other change as well too. But these make no change at all. Most other changes would have either a positive effect or maybe a negative effect. But so this is a little bit of a discussion about what happens with that and what's happening, and I call it running in place. And this is about the conservative nature of natural selection. Okay, it runs in place. If there's 64 possible triplets like we talked about, the ACG, the TTC, 4 times 4 times 4, there's 64 possible triplets. For any triplet, there are 3 possible changes at each base. There's the first one could change in 3 ways, right? The A could become a C, a G, or a T. The next one could have 3 possible changes to any of the other 3 nucleotides, and the last one could. So there's 9 total changes for each of those 64 triplets. So this is just a little bit of math here. So if there's 64 triplets and there's 9 changes that could happen on each of those triplets, that means there's 576 different possible random mutations, okay? And any one of those things could happen. A CGT could become a CGA. That's one of 576. But there's 576 possible. If you look at the genetic code, it reveals that 135 mutations have no impact. That's like I just was talking about with the leucine, right? There were 6 or 5 changes there out of the 6 that 5 changes had no impact at all, right? So there's a total, if you look at all the codes, there's 135 mutations that have no impact. Well, 135 out of 576 is 23%. So 23% of the time that there's a mutation in one nucleotide, it has no impact. It's called synonymous, okay? That means what's going to happen with the other 77%? It's got to have a change, right? So 77% are non-synonymous. That's just what's left over from 100%. Well, it's roughly 77 to 23%. It's roughly 1 to 3, 23 to 77. So if you see that if you would just assume that there was a change in one nucleotide, it would have an impact, a non-synonymous impact, 77% of the time, or 3 to 1 ratio, right? Okay, that's just the math of it all. But in nature, we find a ratio synonymous to non-synonymous. So if you go out there and actually look at what occurs in nature, you find a living creature and you look at what's going on with it, right? And you look in the cells and you see what happened with this thing, you find that there are synonymous ratio to non-synonymous of roughly 3 to 1. So most changes that you see when you look inside an actual creature are much more non-changing, right? They don't change anything. They're synonymous. They have no impact. Well, why would that be? If the math is there, math is math. How could it be that when you look at living creatures, it happens so much more that the changes that are mutating, that are occurring inside them, aren't causing the change in the amino acid that gets made? And back, the, the mutated cells continue. The mutated, I didn't hear.
Is it because the mutated cell doesn't continue to divide?
A mutated cell may not continue to divide, you're saying, right? Would you expect a synonymous cell to be able to divide and keep on going on and have life? Probably 99% of the time, 100% of the time, right? It had no change on it, so it's a living cell. It's been around for, millions of years, billions of years. So, and a synonymous change doesn't have an impact by definition on the cell, so a cell is going to reproduce and survive. What about a non-synonymous change? What about a change that makes a change? Are, are those changes going to be good?
Maybe.
Are the
Might.
changes going to be bad?
Could.
The only changes that are going to survive, probably, or changes that are good are going to survive, and bad changes may have an effect that the creature never gets born, right? That, or it could have an effect that makes it have a shorter life or different things, but maybe you can still sample certain ones that are non-synonymous changes, but it impacts its capacity to survive. It's not valuable. Okay. So in nature, we do find this relationship, which is the reason why I guess the question becomes, this is a tenfold more common end result of no change than change. And why? There's a phrase that answers why.
Ta-da.
It's a proof of natural selection, right? It's a proof that the fittest survive. The things that are most able to adapt, most able to go on with their way of life, most able to fit in with the needs of their community, of their environment, are going to survive, right? So something that has no changes to it is already fit for its environment, whereas something that has a change to it is going to potentially have capacity to fit into that environment, but more often than not, it will not, right? So you see a ratio of almost 10 times as many synonymous changes occur than what you'd consider as possible just mathematically. All right. Is this, I don't know, I think people are following along, but I can delude myself too. I think am I over the top, or you're all right? People are okay with it? Okay. All right, good. Yeah, thumbs up. Here's a question, John.
I think this is about that. So you say we observe 3 to 1 in when we find animals or plants in actual real world. That is, their being there for us to observe them is a result of the changes that have happened being adaptive, or at least for them to either be a non-impacting change or a potentially a, a, an impacting change that doesn't reduce their chances to survive and reproduce.
Right.
So whereas so that's what explains the flip, right? It's not just that. It's the fact of eons of these things continuing to exist and then and reproduce. For those genes continuing to be passed forward.
Right. It's got characteristics. It's got capacities. Every living thing that's made it to this point, we're a product of billions of years of evolution, right? And so we've got features built in all of us, and all living things do, that have evolved for that length of time, that fit us, that make us fit for the environment that we're in. Now, the environment can change. There can be a whole bunch of variety of things that go on, but that can, if the environment changes, we may not be fit for that anymore. I've got some real interesting stories about ice fish, um, that evolved to live in the uh, South Antarctic Ocean, right? These guys have no red blood cells in them anymore. How can it be that a living creature cannot? They need to carry oxygen. Oxygen is what causes respiration and and gives us the energy to do things. Gives the ice fish the energy. So why is it that they don't need red blood cells anymore? Okay, and the answer is, first of all, that very, very cold waters carry more oxygen. Okay, so they're living in an oxygen-rich environment, and their gills can still get oxygen, and their skin is thin enough that it allows oxygen to get to osmose into its system, so it can live. It's evolved in that way. It had mutations that got rid of its red blood cells, and mutations that caused it to have um, antifreeze in its system to pour through to stop crystallization, ice crystals to form, and it can live in that environment. Okay, so it went through these mutations that we're talking about. It's fit for that environment now. But now, as global warming comes, we're seeing the Antarctic waters are warming up by 2, 3, 4 degrees. As those keep going, how easy will it be for an ice fish to regain red blood cells and to get its oxygen that it needs when it's in a less oxygen rich environment and all these things? Most of these things are one-way passages, right? It lost the ability because it didn't need it, but that's different than needing an ability and gaining it, right? And ice fish are very, very susceptible to the warming oceans. They will not be able to evolve a change that does it. So, question?
Yeah, sorry, this is probably off topic, but I just intrigued. So the ice fish, does their blood have free hemoglobin, uh, or do they have, or are they transporting oxygen just suspended the in the water in the blood plasma?
They don't have any replacement for hemoglobin. They can transport oxygen that they can get oxygen to all of their cells just because they live in an oxygen rich environment. Their gills are adapted to even get more oxygen out, but they cannot live unless they're in a very cold environment. And yeah, it's an amazing thing. Yeah,
So you also mentioned direct oxygen absorption through the skin. How large do these fish get?
they get pretty good size, I think. Yeah, they're part of the fishery. They've been fished for human consumption down there, and now they've had their problems with loss of habitat and loss of environment and being overfished and so on too. So, all right, Mike. Let's wait a second, Joanne. Mike's here. This is an announcement. We are not going to get through my whole talk today, but 2 weeks from now we'll continue it. Hopefully, you're finding it interesting.
This process that you've just been describing here, is this the technology behind those tests that sometimes we get? Like, what is the chance of how bad your cancer can be by taking this genomic test?
Hmm. And a genomic test. It's somewhat new, and people are trying this and thinking that you can kind of focus a proper treatment when you see what it is. The things that are going on now that they're doing is they're seeing from the genome that things in terms of, for example, sickle cell anemia, and a genomic test can tell that. There's another disease that they just cleared for treatment with genome replacement therapy and so on. I mean, there's things involved, and I'm not a doctor to say for sure, But I mean, some of this is stem cell stuff too, where in other countries, I think they're putting stem cells in people to try to rebuild that the stem cells can create the proteins and have the proper genomic structure. But I know that this then leads into what they're looking at here, and what the recent Nobel Prize was for the CRISPR techniques, right? Where if you find that someone is missing a certain thing in their genome, like the thing that makes proper hemoglobin, and that's what's missing in sickle cell anemia patients, you can then add, with CRISPR techniques, you can add into their genome the, the gene for creating hemoglobin. And they've had some amazing results just in the last 3 or 4 years. I know I gave a full talk about CRISPR, I don't know, 3 years ago or something like that, but Jennifer Doudna, and fantastic capacities for addressing certain diseases in that way. And it also brings up certain questions, like if you find that you can fix someone's lack of having hemoglobin, what about if you were a basketball player, you really want your kid to be a basketball player, and you find that they have a genome that's going to make them be 5'11" or something like that, that you can then start doing it for odd purposes, and who's going to get money, the backing, and there's a lot of moral questions involved with gene splicing and so on.
Wasn't the gene therapy for sickle cell just approved?
Yes, yes. There's that and one more are both approved. They've been doing tests and trials
Multiple
for the last
sclerosis,
3 or more years.
Is it multiple sclerosis that they're doing now?
It was a disease they mentioned that it didn't ring a bell with me, so I don't think that's the one I heard. It was some other disease, but again, that could be easily Googled and found out as well too. All right, so we're talking a little bit more about evolution, which is good. This is another level of genetic structure. Over the course of millions of years, many genes show very little change. Here's a list of a bunch of different genes. These are codes for the different amino acids that make a gene, and a thing called the elongation factor. And the elongation factor is something that's in every cell, that when it goes through mitosis, it unwraps and elongates the chromosome, the DNA, so that it can travel through the ribosome in the cell properly and then get replicated. So your cell has this elongation factor, tomatoes have it, all these different creatures have it. So this is the code for humans. Each of those stands for a different amino acid. This is for a tomato. The last letter went from a V to an I for tomato. 26 amino acids there, and one change between a tomato and a human. Think of the times how long it's been since a tomato and a human had a common ancestor. It's nobody in your family book, at home. Okay? And that's the one change. It's a pretty good indication that we, first of all, do have a common ancestor, right? And to me, it just can be very amazing that we and tomatoes can be said to have a common ancestor, right? My wife probably thinks it's less common. Yeast. Yeast has only one more change. The fourth one back from the end. How close we are to yeast. Archaea. There's about five further changes in archaea. Bacteria, from 3.5 billion years ago, we split off from them. 6 further changes. So now scientists look at, well, which ones have stayed the same? Which ones are immortal genes? Which ones have been with every living thing for 4 billion years? There's 14 of them there, over half. So this was something that processed and made the first cells operate well, right? And so that gets passed on. That doesn't mean that there are only 14 or 12 times that there was a mutation that occurred, right? But whenever a mutation occurred, mutations occur randomly and often, relatively. But if it doesn't help the thing survive, it dies off, right? The ones that maintain are the ones that are critical that help it survive. There may be slight adaptations and things are done a little bit differently. Some are changes that had very little impact or no impact or a positive impact, right? But all those things are an indication, a support of evolution. Okay. But how do they do that? Do they go through every gene? Think of this 6.5 feet of genes, all this gene. How do they go and how do they actually do a gene to see how close things are? Do they do every gene in a human? They don't. They look for special things that work in. These are called signs, short interspersed elements. And these are things that you can find in most creatures that come in, and once they're there, they're interpositions in the genetic strain. They're not that necessarily doing something. You don't even know what they do. They could have been what was usually called or formerly called junk DNA. But if you look, this is testing, and you get, when they test for locations of things, they look at wavelengths and what's there. And across the top, you see a whole bunch of different species named human, bonobo, chimpanzee, gorilla, orangutan, siamang, a green monkey, and an owl monkey. An owl monkey is a nocturnal animal. And these are markers for coded structures inside their body. If you look at this one at the bottom, those two, the owl monkey and the green monkey, are not showing that short interspersed segment there, right? As you go up to look at another area, so it shows that we're more common with siamangs, orangutans, gorillas, chimps, bonobos, etc., and humans. And as you work your way up, other things drop off. You're not seeing other signs that are there. You see at level C, we have similar signs with bonobo, chimps, and gorillas, but not with orangutans, siamangs, green monkeys, and owl monkeys. The next one, we have a sign that's common with humans, bonobos, and chimps. And finally, there's a sign at the top that's only common to us. Okay? So this is a pretty amazing way to be able to create a structure. I'll go through a couple. This is a siamang, if you don't know. They have this pouch that expands and they can be very loud. Interesting. So we're similar to them. You see similarities. And based on these signs, we can do a very straightforward tree of life that shows when, this really should say an evolutionary tree of primates, not hominids, I'd say, but human, chimpanzee, bonobo, gorilla, and it shows when things have split off and at different times and why. So these trees were made long ago. Darwin had a famous tree in his book, right? But that tree was usually done by looking at kind of similarities. And sometimes it's hard to see similarities, and it may not be all equally viewed by different scientists. But this is a straightforward way to evaluate. All right. I've got a lot more to talk about genes and evolutionary patterns to them, but I'll stop now, and any questions or thoughts are welcome to take. Thank you. Oops, went by.
So on the the different nucleotide substitutions, you said that certain substitutions are statistically more prevalent in actual observations in live cells. That gets me to questioning: is there, or do we know of any reason why a particular codon is associated with a particular amino acid? Is it random arbitrary, or is that the evolutionary advantage in some very early life form, that a particular a particular codon or a particular pair of codons that are more likely to be substituted should code for the same amino acid?
Well, it seems like the amino acids that have pairs seem to have like one small codon change, or in the case of leucine, two. Most of them only have, some of them only have one expression. And uh, it's hard to say exactly why. I guess if it doesn't code for anything, then it's a stop code. And why one codes for something and something slightly different codes for the same thing, I don't think I'm probably able to say why that is. It's either, I don't know who knows why, or, it's just a feature of the situation, I guess.
Mike, if you look at the machinery for how proteins get strung together, that's the machinery of the ribosome. And the ribosome eats these codons, three bases at a time. It eats those, and then it has the ability to grab a matching amino acid out of the cytoplasm and start stringing that together. So it's a coevolutionary progression that has the literal machinery of the ribosome, which is my favorite organelle. I don't know if you guys have a favorite organelle.
It's up there.
Yeah.
Yeah.
The ribosome has evolved in such a way that it grabs a particular amino acid based upon a subset of particular codes. Now, why it expressed that in terms of its geometry and the abilities it has as a machine, I'm not sure that there's a definitive answer other than random chance, but maybe there's a biochemical. Maybe it has to do with particular energy levels of the different codons, et cetera.
Mm-hmm. Yeah.
Very detailed, I'm sure.
Right. Yeah. And I'm sure people are looking at that and seeing how you can produce what you want to produce. But, question?
Said the ice fish may or may not be able to develop hemoglobin, for instance, when things get warmer. Why not? I mean, the mutations change at a constant rate. The mutation doesn't know what happened in the past. It's just making changes. And some change that could develop a hemoglobin type chemical in its blood could happen again, right?
Mm-hmm.
You don't know.
Right. I would say that scientists look at it as so unlikely because you have a capacity, and it's easier to, you'll admit, it's easier to lose a capacity than it is to gain one. I think it has to be seen that I would say that there have been common developments of, for example, vision, right? Uh, in caves, fish have not had vision and they've developed vision if they've brought out. But I would say that there's been, the octopus has developed vision. That there's no doubt that there's potential for development of any type of capacity, right? But I think I'd ask you to agree that it's easier to lose a capacity than it is to gain one. And because it's shown so often that, for example, animals that live in the depths of the sea lose color vision, right? if you, the whole, I mean, Darwin's whole thing was if you don't have, if you lose it. If you don't use it, you lose it. If you don't use a capacity, you're going to lose it. Fish that go into a cave become blind. So there's no question in my mind, you know, and I can probably come up with a lot more examples, but there's no question in my mind that it's much more common to lose an unused capability than it is to develop a needed one. There's nothing in evolutionary thought that says, I know how to compensate for this thing that is going to come in the next 50, 100 years. Right. Right. But but the ice fish had a gradual adjustment as temperatures got slowly colder and colder through geological time. Climate change is not a geological process. It's a man-made process that's occurring at much faster rate. And, and the capacity of these fish, can they adapt to it? Is it possible? Yeah, evolution makes people like us out of tiny one-celled organisms. So yes, things can happen. But I would say that scientists are very leery. I mean, 99% or more of creatures that have ever lived on Earth are extinct, right? The common thing is if you aren't fit for your environment, you're going to die out and something more fit is going to take your place. So those are my answers to you. And you're cherry picking because you're picking an ice fish, which was a successful adaptation that succeeded. And you're ignoring the 99% of other fish or different species that weren't able to adapt to those changes and have already died out in history, right? So, no, I think it's a really interesting point, and it gets me on my toes to have to define and defend it. But yeah, yeah. But no, I think it's a great question. But go ahead. Go ahead.
Just a quick question on a more basic level. So all this replication that's going on, what percent ends up being a mutation?
Mutations are interesting things because you have to define, scientists say, as to exactly what makes a mutation. I think Dave has a T-shirt on that says something, and we've all heard it. We're like 99.5%. He's got 98% chimp. You know, and when you look at what that is, that scientists are looking at that in a certain way. What makes something be a change? You can have all these changes that change leucine in it. You could find with genomic testing nowadays, you could find 17 changes in a species that didn't change it at all. They'd be, you know, virtually looking the same. If you go down to that codon level and say, these are 17 different codons in that is, and you could see times where whole strings, like hundreds of characters or thousands of characters, are repeated, right? Or sometimes they're reversed or they're mirrored. And that all occurs in terms of mutations. And is that one mutation? Is that, you know, 10,000 mutations? And if it doesn't do anything, is it no mutations? So it's a very dicey topic. And when you say 98% are the same, they are actually throwing out all those duplications. They're tossing out the fact that two chromosomes pairs that were in chimps bonded together to make one chromosome pair in humans. You know, I mean, it's a question of definition almost of how do you really define of what is a mutation, you know? And a mutation that didn't change anything, is that as critical as a whole series of things? Or so
Is that
it's
known as a change, which is no change?
right. Yeah. Is a change if it doesn't change anything? Exactly. I mean, if it still codes for the same amino acid, I think I wouldn't go along with that, but it maybe allows for that step. And that's what they've seen when they've done these tests at Michigan State University with like generations and generations of bacteria that they wash and refeed every week or something like that. And they've got like 10,000 or 10 million generations of this bacteria. And finally, it can start eating a different source. You know, they feed it to the extent it starts starving. It goes through a die off every time they refeed it, and then the strongest survive. It's kind of an evolutionary task that they're doing. Fascinating stuff. Right, right, yeah. I mean, just the fact, and we'll talk about that more. When genes duplicate, that allows things like three-color vision. We're done, right? You're not taking any more. Okay. If I lose my hearing and my eyesight improves, huh? Could you say that again? No. I think that's adaptations, right? That's your body adapting to solve your world problems, to solve your way of evaluating the world, how best you can thrive in the world by using what resources you have. Right. And you wouldn't pass that on. That's somewhat Lamarckian. All right. Fascinating stuff. Thanks for